you. My name is Akbar Vaseghi. I come from Iran, and I would like to talk about my one. Actually, it is this title is called "Nanoparticle for Biomedicine Application." It's a review for and collection 2 and 3 my working project in Iran, and I think it is good for you. This slide shows the outlines, my introduction, and my lecture, including the 3-part introduction, a gold nanoparticle, diagnosis and imaging, therapeutics purpose, and the discussion and conclusion and discussion. I'm strong inclined believe that the future of the world will be effective on only the serene technology, and recently in some scientists believe the 40-part biotechnology, nanotechnology, information technology, and I forget cognitive. The 4 parts for the scientists, and nanotechnology is a crossroads in biotechnology and nanoscience, and I think is after cancer decay technology for 21st century. This is the 2 men. It's a pioneer for the nanotechnology, and you can know it's a fireman, and the Americans using gold as a nanotechnology, particularly gold nanoparticle for a medicine application. A gold nanoprobe and gold nano actually the uses of the gold of medicine data back is 2000 BC, and I think the China and Egyptian, and another application is the gold for the different purpose in medicine. You can search in the slide showed increases the using and application, and another purpose in gold nanoparticle. You can see 70,000 paper application 2011, and this slide shows another application is gold nanoparticle, particularly in gold nanoprobe for a detection in medicine. And the slide shows another aspect of using a gold nanoparticle. This shape is a different shape as gold nano. For example, gold nanorod, braid, gold nanoparticle, and all them using for a biomedicine application for a different purpose. But why the gold nanoparticle is attractive for a scientist for a different purpose? There are a variety of region that a gold nanoparticle is very attractive for biomedicine application. For example, in the easy for a synthesis process, unique and the special optical purpose, and a high surface, and some another characterized as a very good application. This slide shows one of the model for the gold nanorod that using in the cancer therapy for light therapy. This slide lists the different way of the application of gold nanoparticle, nanobiosensor diagnosis and treatment for disease, identification and treatment, drug and gene delivery, gene analysis, virus study, and so on, and some another. I think it is one of the famous paper that described it with American in 1996 in using gold nanoparticle with hybridization of gold in DNA and some biological. This slide shows the 3 methods for the design of the gold nanoprobe for the detection in the some virus or some genes that's very important for how can I use them. And this is a slide show my design for the application. You can when you search in in application of gold nanoprobe for detection of some special genes, only you can see as these models for the detection, because this light is shorted and easy for detection. And when the light is longer, it's very hard for detection, because a gold nanoparticle cannot aggregation and for some purpose. We designed after 2 probe for a detection of some bacteria for a detection is these models, and you can see we aggregated this nanoparticle in the genomics DNA, and it's very hard for a detection, because the aggregate is very hard, and detection was hard for particular when we design is the colorimetrical detection is very hard. And this slide showed medicine application in gold nanoparticle from dot plate and some another purpose for gold using a gold nanoparticle. And this slide shows one example in using a gold nanoparticle for CT scans, MRI for detection with a combination and some dendrism for using a high level observation. So far is another example for, but it is a slide show my result for a detection in bacteria with special designed and aggregated in gold nanoparticle in the genomics, you can see it's a different when after and before aggregated, and you can see it's different and shift wavelengths in before and after the using for special genomics in bacteria, and the figure 3 shows some negative control for my design. And this slide shows specifically for hybridization gold nanoparticle for in this design. And this slide shows especially for specifically for my design for gold nanoparticle, but when we use it in the three design for a gold nanoparticle, it is we saw the variety of the result. That's a negative result and positive result for is not good for me, because I designed and some special region for our genes, but this slide shows the variety of the result in some nanodrop. And the slide showed like them it's sensitivity for after hybridization, and we published in my result in journals, and okay. And the slide shows the therapeutic purpose for a gold nanoparticle. You can see increase of the for a gold nanorod, particularly gold nanorod for the phototherapy for and application, for example, in cancer therapy, and or another using for a drug, because the gold nanoparticle can have a special characterization for the targeting some issue in body. And this slide shows some records for the using a gold nanoparticle and some different region. And this slide show in my design for the using some special peptide for a nanoparticle when attach a nanoparticle for a phototherapy and some another purpose. You can see when we attach a gold nanorod with peptide and using some different legs for the carbon and the CO2 for the using attach a gold nanorod and the peptide for detection. And this slide show in some design for a beginning for the some one small chips for detection. This slide show in the conclusion only. I'm sorry for because this slide and this present for reporting we are working. And the end, I'm thankful for my professor and you for attention. Thank you very much. Do you have any question for me? Characterization? Okay. Characterization gold nanoparticle with any different of the biological, for example, in protein and DNA, RNA, and peptide particular. And recently, it's very attractive for scientists that working in peptide for targeting, because have some special characterization for targeting in some, for example, in cancer cells and some another disease. And when we characterization with DNA and the gold nanoparticle, we're using some life in carbon, because all the majority of the paper describe it in the 6 or 4 or 5 carbon for using it in attach. But we use it and often, for example, in 8, 9 particular, someone when we conclude it, when conclude it is in the gold nanoprobe, we use it after 15 carbon for after 30, because and some topological approaches, it is very hard for detection, and it is a very important section for the this design for gold nanoparticle, and we use it after 15 carbon for the conclusion is gold nanoparticle. You're welcome. Another question? Any question? That's good. Thank you. Thank you very much.